For almost the entire history of our species, human beings slept in the dark. Not the darkness of a bedroom with curtains drawn and a phone screen glowing on the nightstand. Not the partial darkness of a modern city where streetlights filter through windows and the sky above is never fully black. The darkness of a night with no artificial light source anywhere in the landscape. A darkness so complete that holding your hand in front of your face produced nothing. No outline, no shadow, nothing but the undifferentiated texture of the dark itself. This was the condition under which the human sleep system evolved. For hundreds of thousands of years, the signal that it was time to sleep was not fatigue alone, not the feeling of tiredness that accumulates over the course of a waking day, but the disappearance of light. And the signal that it was time to wake was not an alarm, but the gradual reappearance of light on the horizon. The relationship between light and sleep in human beings is not simply a matter of convenience or habit. It is one of the most precisely engineered biological systems in the human body, a system so sensitive and so finely calibrated that it can detect light at levels far below what the eye needs for vision, and that responds to light and darkness by regulating not just when we sleep, but what happens to virtually every organ in the body while we do. Understanding this system, what it does, how it evolved, and what happens when the conditions it was designed for are no longer present, turns out to be one of the most important stories in modern medicine, because the story connects artificial light, which human beings have been using in increasingly bright and persistent forms for only the past few centuries, to a cascade of biological disruptions whose consequences we are only beginning to fully understand. Let's begin with the biology. The neuroscientist Joseph Takahashi at UT Southwestern Medical Center has spent decades studying the molecular mechanisms of the circadian clock, the internal timekeeping system that regulates the daily rhythms of virtually every cell in the human body. Takahashi's work, for which he shared major prizes in chronobiology, revealed that the circadian clock is not simply a system in the brain, it is a property of individual cells throughout the body, encoded in gene expression cycles that repeat with a period of approximately 24 hours, and that govern when cells perform which functions. Every cell in the body has a clock. The liver cell runs its metabolic functions on a schedule. The immune cell regulates its inflammatory responses on a schedule. The heart cell modulates its contractility on a schedule. The gut cell manages its digestive functions on a schedule. The cancer-suppressing genes in every cell cycle through their activity on a schedule. All of these cellular clocks are coordinated by a master clock, located in a region of the hypothalamus called the suprachiasmatic nucleus, abbreviated SCN. The suprachiasmatic nucleus receives direct input from a specialized class of photoreceptors in the retina, cells that are distinct from the rods and cones that enable ordinary vision, called intrinsically photosensitive retinal ganglion cells, or ipRGCs. These cells were only discovered in the early 2000s by the neuroscientist David Berson at Brown University, and their discovery revolutionized our understanding of how light affects the human body. The ipRGCs contain a photopigment called melanopsin, which is particularly sensitive to short-wavelength blue light in the range of approximately 480 nanometers. When ipRGCs are activated by this wavelength of light, they transmit signals directly to the suprachiasmatic nucleus, which interprets this signal as indicating that it is daytime. The SCN then synchronizes all of the body's cellular clocks to this interpretation, setting the phase of the circadian rhythm to align with the external light-dark cycle. This system is what chronobiologists call the primary zeitgeber, a German word meaning time giver, for the human circadian clock. Light is the most powerful signal available to the biological clock for synchronizing its approximately 24-hour rhythm with the actual 24-hour cycle of the Earth's rotation. The downstream effects of the suprachiasmatic nucleus's response to light are profound and far-reaching. One of the most important is the regulation of melatonin secretion by the pineal gland. Melatonin is a hormone that serves as a chemical signal of darkness. The pineal gland begins secreting melatonin in the evening, typically a few hours before habitual sleep time, reaches peak secretion in the middle of the night, and suppresses secretion in the early morning hours. This melatonin rhythm signals to the body's tissues that it is nighttime and prepares them for the physiological processes that occur during sleep. What most people do not know is that melatonin secretion is exquisitely sensitive to light exposure. Exposure to light, particularly blue-spectrum light, during the hours when melatonin would normally be rising, suppresses melatonin secretion almost entirely. The light threshold for this effect is remarkably low. Studies have found that exposure to ordinary room light in the evening, at intensities easily produced by standard indoor lighting, can suppress melatonin by 30 to 60% compared to dim light conditions. This is the core of the problem. For almost the entire history of our species, the only light source available after sunset was fire. Firelight, campfire or candle or oil lamp, is warm spectrum light with very little blue content and considerably lower intensity than daylight. The circadian system, which is most sensitive to blue spectrum light, was barely affected by firelight. Our ancestors could sit around a campfire in the evening without significantly disrupting their melatonin rhythms, because the light from the fire was not the kind of light that the ipRGCs were most sensitive to. The electric light changed everything. Standard incandescent bulbs, and much more dramatically the LED lighting that now dominates indoor environments, produce substantial amounts of blue spectrum light. The screens of smartphones, tablets, laptops, and televisions produce particularly high concentrations of blue light directly at the wavelengths to which melanopsin is most sensitive. And these light sources are used not just in the morning or the afternoon, but in the hours immediately before sleep, at precisely the time when melatonin would normally be rising and the circadian system would be signaling to the body that night has arrived. The sleep researcher Matthew Walker at the University of California, Berkeley, whose 2017 book, Why We Sleep, became one of the most widely discussed works in popular science, has documented the consequences of light-induced melatonin suppression in extensive detail. Walker's synthesis of the existing research found that exposure to LED light in the evening can delay the onset of melatonin secretion by up to 3 hours, shifting the effective biological night by 3 hours and producing a corresponding shift in sleepiness. The person who uses a smartphone until midnight is not simply staying up later. They are biologically misaligning their sleep timing in a way that has consequences for every physiological system that runs on the circadian schedule. The consequences of circadian disruption extend far beyond sleepiness. The chronobiologist Till Roenneberg at Ludwig Maximilian University of Munich coined the term social jet lag to describe the chronic misalignment between the body's biological clock and the schedules imposed by modern social life, including work, school, and artificial lighting. Roenneberg's research has found that social jet lag is associated with elevated rates of obesity, metabolic disorders, cardiovascular disease, depression, and impaired cognitive function. The mechanism through which circadian disruption produces these health effects connects to the fundamental biology of the cellular clock. When the master clock in the suprachiasmatic nucleus is receiving light signals at the wrong time, the cellular clocks throughout the body lose their synchronization with each other and with the external world. The liver is running its metabolic programs on one schedule, while the gut is running its digestive programs on another, and the immune system is running its inflammatory programs on a third. This desynchronization produces physiological inefficiency and, over time, contributes to the development of chronic disease. The epidemiological evidence for the health consequences of circadian disruption has become increasingly compelling over the past 2 decades. Shift workers, who are chronically exposed to light at night and dark during the day, show significantly elevated rates of breast cancer, colorectal cancer, prostate cancer, type 2 diabetes, cardiovascular disease, and metabolic syndrome compared to day workers. The World Health Organization classified night shift work as a probable human carcinogen in 2007. Based on epidemiological evidence and on the known mechanisms connecting light at night to the suppression of melatonin and the disruption of circadian clock genes that regulate cell division. The relationship between light at night and cancer risk has been a particularly active area of research, connecting the circadian biology to the fundamental biology of tumor suppression. The circadian clock genes regulate a range of tumor suppressor pathways, and their disruption through chronic light-at-night exposure appears to create conditions that are more permissive for tumor initiation and progression. The oncologist David Blask at Tulane University Medical Center has conducted research demonstrating that melatonin directly suppresses the growth of certain tumor types, and that the suppression of melatonin by light at night removes this suppression and accelerates tumor growth. The evolutionary perspective on why the human body is so profoundly affected by light at night connects to the timing of the original problem. For the entire period of human evolutionary history, light at night simply did not exist in any form that could affect the circadian system. The only natural sources of light at night were the moon and the stars, and neither of these sources produces light at intensities or at wavelengths that significantly activate the melanopsin system. The biological clock evolved in a world in which the presence of light reliably indicated day and the absence of light reliably indicated night. The reliability of this signal was essentially absolute. The cognitive neuroscientist Charles Czeisler at Harvard Medical School, who has spent 4 decades studying the relationship between light, circadian rhythms, and health, has argued that the introduction of artificial light after sunset is one of the largest experiments ever conducted on the human species, an experiment that has been running for only a small fraction of human evolutionary history and whose full consequences are still being measured. Czeisler's research has documented some of the most striking consequences of light-at-night exposure. In a landmark study conducted at Harvard, Czeisler and his colleagues kept research participants in an environment with controlled light exposure for several weeks, and found that even a single week of camping with no artificial light exposure, relying solely on natural sunlight and campfire, was sufficient to advance the biological clock by approximately 2 hours and to significantly improve sleep quality and daytime alertness. The body's circadian clock responded rapidly to conditions resembling those under which it evolved, and the consequences for sleep and waking function were measurable and substantial. The practical implications of this biology have generated a significant and growing literature on what is sometimes called sleep hygiene, strategies for aligning behavior with the body's biological requirements. The most evidence-based of these strategies center on light management, reducing exposure to blue-spectrum light in the evening, increasing exposure to bright light in the morning, and creating sleeping environments that are as dark as possible. The epidemiologist Ricardo Orozco at the University of Southern California has studied the relationship between bedroom darkness and metabolic health across large populations, and found that people who sleep in darker rooms show significantly better metabolic outcomes than people who sleep in rooms with ambient light, even after controlling for total sleep duration and other confounding variables. The quality of darkness during sleep appears to matter for health outcomes independently of the quantity of sleep. What the science of light and sleep ultimately reveals is a story about evolutionary mismatch, about the consequences of placing a biological system in conditions radically different from those in which it evolved. The human circadian system is not simply a clock that tells you when to feel sleepy. It is a fundamental organizational principle of the body's biology, coordinating the timing of metabolic, immune, cardiovascular, reproductive, and cellular repair functions across every tissue in the body. Its design assumes that light means day and darkness means night, with sharp transitions between the two occurring at predictable times. The modern environment violates virtually every one of these assumptions, and the consequences of that violation are not simply that people feel tired. They are written in the body's biology, in cellular clocks running out of synchrony with each other and with the world, in melatonin rhythms shifted by hours from their evolved timing, in tumor suppressor genes cycling through their programs at the wrong time of day. The darkness that your ancestors slept in was not simply the absence of light. It was the condition under which your body learned to repair itself. And the blue glow of the screen you checked before you went to sleep last night told every cell in your body that it was still afternoon. Thank you for watching, and I will see you in the next video. If you find this video interesting, please consider subscribing for more content like this.